It is my pleasure to welcome you all to our Zoom lecture today, learning about the COVID-19 and pet study at the University of Washington. Our speaker today is Julianne Meissner, a veterinarian, epidemiologist, and clinical assistant professor in the Department of Environmental and Occupational Health Sciences in the School of Public Health. Julianne's research and teaching focuses on health at the human-animal environment interface and statistical and epidemiologic methods suitable to addressing health threats at that interface. We also have with us today Vicki Ramirez, a research coordinator at the Center for One Health Research in the School of Public Health, who is also a member of the CAPS project team. So lots of knowledge here with us today to answer any questions you have. Go ahead and throw those in the chat throughout, save them for the end as well, and you can raise your hand. We'll have time for questions. As a reminder, we are recording. We look forward to joining you throughout the session. Thanks so much, Julianne, for being here, and I'm going to pass it off to you.
Thanks, everyone. I'm just going to get my screen sharing underway. Talking today about our CAP study, but before we get there, I want to talk to you a little bit about One Health in general. And perhaps those of you joining are joining because you are already interested in One Health. But if not, I hope if you are, that gives you some more background. And if not, then hopefully this provides you with the context you need to understand what a One Health study involves and how our COVID and pet study is a One Health study. So first, human, animal, and environmental health have traditionally taken a siloed approach to handling human, animal, and environmental health, both in the research that we do to address health threats in these sectors, but also in the way that we administer health. So human health is typically the mandate of a Department of Health, while animal health is at the Department of Agriculture, and environmental health is Department of Ecology or Fish and Wildlife. And here in Washington State, you probably know that the medical school and the School of Public Health are here at the University of Washington in Seattle, but the veterinary school is all the way out in Pullman. So in the US, that's pretty standard that medical schools and veterinary schools are actually geographically distinct, the new medical school at Wazoo notwithstanding. But this siloed approach doesn't allow us to investigate health threats that blend over between these interfaces. And today I'm going to be mostly talking about zoonoses, so diseases that go from animals to humans, and I'll define that in a bit more detail in a minute. But the health threats that exist at this human-animal environmental interface go far beyond infectious diseases to environmental hazards like heavy metal exposure, carcinogens, things like that, and some social behavioral health threats as well, and opportunities for health promotion that cross this interface. So the One Health conceptual framework recognizes that this siloed approach is counterproductive and allows things that sort of cross over these interfaces to fall through the gaps, and instead takes a holistic view and considers all three of these sectors together. So some key terms, just to make sure we're on the same page. I mentioned zoonosis in passing. A zoonosis is a disease that goes from animals to humans. A reverse zoonosis is the opposite. It goes from humans to animals, although in common parlance, we often just use the term zoonosis to refer to something going back and forth. The last two terms aren't actually that relevant to this presentation, but they're just in there to highlight the fact that veterinary epidemiology is a discipline in its own right. So on the veterinary side of things, an enzootic is the term that we use instead of an epidemic, and an epizootic is the term that we use instead of an epidemic. And at the bottom, this is actually a headline from the Point Defiance Zoo in Tacoma, but this is an example of a reverse zoonosis with an elephant at the zoo testing positive for tuberculosis, and that elephant most likely was infected from a zookeeper. That elephant isn't really going out and exposing itself to anybody except for the animals that take care, or the humans, I'm sorry, that take care of them. So now that we've got that background, I just want to talk a little bit about zoonotic disease emergence. This is a pretty outdated map. MERS isn't on here, Zika isn't on here, and COVID-19 certainly is not, but the point remains, and that is that the majority of the diseases that are going to, like COVID-19, like Zika before it, MERS and SARS before it, suddenly come out of seemingly nowhere and make us all quite concerned for our health, are most likely going to come from an animal. And you can see this is happening all over the world. And what I want to draw your attention to here as well is these purple stars. These are diseases that come from bats. I'm going to talk about bats in a little bit more detail later on. But while these, COVID-19, the SARS, the MERS, they are very scary. They grind our economies to a halt. they make a lot of news. These are actually not the zoonotic diseases that have the greatest burden globally. Those are the bacteria and the parasites and the protozoa that quietly every single day spill over from animals into humans, and infecting them, and in many cases causing chronic illness. So this is a slightly older report, but about 1 billion cases of illness and millions of deaths each year are caused by diseases that come originally from animals. And the majority of these, again, are not those really scary viruses, as scary as those viruses are. So why does this happen? I'm going to be talking about 3 main drivers of zoonotic disease spillover. And the first is that the human population is growing and expanding into new geographic areas. So this is we all know the human population is increasing. It's depicted here as a linear trend for simplicity. But on that is an exponential increase in poultry populations. And this is because while the majority of that growth in human population is occurring in the developing world, where it is accompanied by economic development. And even though a lot of us may be vegan or vegetarian, or know people who are vegan or vegetarian, generally as populations acquire wealth and develop, they tend to want to consume animal source protein in greater quantities. So what we have going on in the human population, we are expanding outwards. We are also concentrating inwards into cities. This is Delhi, 1992 on the left, 2004 on the right, and the darker red is a more dense population. And to feed these cities of people, we are developing these cities of animals that oftentimes are located somewhat close by to these cities of people. And so this is happening, I talked about the developing world, but it's happening in the US as well. And in Europe, we have a consolidation from a lot of small farms, many small farms with very few animals on them, to much fewer farms with many more animals on them. So going from farms that look like this to farms that look like this. And those of you who are not coming from an agricultural background might immediately think, well, this farm looks awful and I would much rather be on the previous farm. But this type of operation is actually much more able to keep out pathogens. So the previous slide, these pigs, the air is, it's open air, to birds flying overhead that may have avian influenza, to the fences are open to wild boars that could get in here and have nose-to-nose contact with these pigs and could be transmitting Brucella. So both farm types have their pros and their cons, but there is a large scale shift to this type of farm. But one of the big downsides of this type of farm is the environmental implications. So this is a hog operation. I believe it's in North Carolina. On the right there is what is very euphemistically called a lagoon, and it is essentially a pond of pig waste. And that is, these large operations need to manage that waste in some way. And this is a real problem when Hurricane Florence hit a couple of years ago in North Carolina, because it hit an area where there are a lot of hog operations, and you can imagine why you wouldn't want a hurricane, or what the environmental implications could be of a hurricane hitting this lagoon. Okay, so the next factor I'm going to talk about, and this is far from exhaustive, but just 3 that I think are sort of summarize the situation well, is changes in land use and climate. So this human population I mentioned concentrating into cities, but it is also expanding outwards, both for to house those individuals, but also to grow the food that those individuals consume. And so that often in many ecosystems results in deforestation. And when we deforest, when we encroach on those ecosystems, we drive the animals in those ecosystems out, because they now have to look elsewhere for their food. So this is and their water and their habitat. So this is a watering hole. You can see a woman and a young child using this water. You can see cattle using this water. This is an opportunity for those what we call endemic zoonoses I mentioned, those bacteria and those protozoa and those worms that quietly go back and forth to humans all of the time. This is an avenue for them to be transmitted. But I can also assure you that at nighttime, wild animals are probably using this watering source. And in addition to driving humans and their livestock, their domestic animals, into closer proximity with wildlife, additionally, when we disturb ecosystems, we destabilize a balance that's present in those ecosystems, and that can allow sort of winners and losers. So you can have some species that die off and some that become more numerous, resulting in a reduction in diversity. And so any pathogens that are harbored by those species that sort of are the winners there's more of it essentially because there are more hosts. And so more hosts with more pathogen, plus this increased contact I discussed, allows more opportunities for wildlife to contact livestock. And these sort of very scary viruses, the one in that blue picture, the COVIDs and the SARS and the MERS, most domestic animals, like most people, have no pre-existing immune knowledge of those pathogens. So if a spillover event occurs, and a spillover event refers to a zoonotic transmission event, so a situation in which a disease jumps from an animal to a human, or a human or an animal across the species, essentially. So a wild animal to a domestic animal is also spillover. But if you have a spillover event from a wild animal to a domestic animal, oftentimes that domestic animal, let's say a pig or a horse, would get very, very, very sick, and they would shed a lot of virus. And the people that take care of them have a lot more contact with those domestic animals than they do with the wildlife reservoir that initially infected that domestic animal. So that's an effect that we call amplification, and we've seen that with quite a few diseases. And the last, and this is not unique to zoonoses, but international trade and travel have increased. We've all seen this with COVID. That allows diseases to travel across the globe. But what is a little bit unique to zoonoses is it's not just people moving place to place that can transmit diseases, but animals and animal products. And so that could be illegally traded or poached wildlife and wildlife products for the pet industry or as ornamental purpose or for ornamental use. And it can also be agricultural animals. So when you go to the grocery store, there's a reason that you see some countries, but not every country represented in the meat or the eggs aisle. And that reflects in part how adequately international governing organizations feel countries are controlling certain diseases and allowing them to access international trade markets or not if that disease control is not adequate. Okay, so what about COVID-19? So we do not yet know the reservoir host. The reservoir host is the wild animal that originally sort of harbored COVID-19. We don't know what that animal was, nor do we know whether there was or what species served that amplification role. So that that role in which an animal that has closer contact with humans, but is immune naive to COVID-19, to SARS-CoV-2, would get exposed to the reservoir host, get very sick themselves, shed a lot of virus, and infect a human. These pictures, these animals are from SARS-1. We do think that it most likely was a bat for SARS-CoV-2. This bat in the middle here is the reservoir host for SARS-CoV-1. It's a horseshoe bat. There's the masked palm civet is on the right. That was the amplifying host for for SARS-CoV-1. It's possible at this juncture that we are not going to ever know the amplifying host for SARS-CoV-2. So I mentioned I'm going to get back to bats. Bats are the reservoir for a lot of different zoonoses, many, many different zoonoses. We think they're the reservoir for SARS-CoV-2. We know they're the reservoir for SARS-CoV-1. They're the reservoir for MERS, the reservoir for rabies, for Ebola, for lots of things. And I wanted to talk to you briefly more as sort of an interesting set of facts about why bats serve as such fantastic disease reservoirs. And first, again, sort of an interesting series of facts. Bats are divided into micro and megabats. You have on the top right a microbat. This guy is about 1/3 of an ounce. And on the bottom right, you have a megabat. This bat would weigh about 3 ounces. And you can see the difference in the eye versus ear ratio in addition to size. But both micro and megabats roost in large groups, and that allows, as we've all learned the hard way from COVID, when we all get together in really tight and close spaces like a cave, it's very easy to transmit diseases. Bats, location, are the only mammals that fly, and in doing so, they can move over large distances, bringing whatever viruses they harbor with them. So sort of similar. Oh. Did you lose me for a second? Am I back?
We lost you just for a hot sec, but you're back.
Okay, cool. Zoom's your internet connection is unstable. Let me know. So I think we're good. So sort of like those airplanes that take diseases all over the world. They also don't swallow their food. So sort of like my toddler, when particular megabats, they're eating their fruit that they eat, they will chew it up and suck out the juice and then spit out the rest of it. And I should mention that the microbats are the vampire bats are a type of microbat, and the rest of them eat insects, while megabats eat fruit. And that the fact that they don't swallow their food becomes particularly relevant in a moment. And then they both have long lifespans. They can live several decades. And those of you who come from the STI world probably know that as you get older, you acquire more and more viruses. So that's important as well. And then they have some immunological features that allow, they have all these opportunities for to acquire viruses, and then they have particular features to their immune system that allow those viruses to kind of stay there. And one is that when they hibernate, their body, their metabolism really slows down, and they sort of stop fighting those viruses. And the other, to make them able to fly, they have hollow bones, so they don't have any bone marrow, and so they don't have any B cells, and B cells are pretty critical in antibody responses. So I mentioned those fruit that these, the flying foxes, so the fruit bats, the megabats, sort of chew on, suck on, and spit out. And I mentioned earlier the amplifying hosts, so those domestic animals or could be wild animals as well that humans contact through markets, which is possibly what happened in SARS-CoV-2. But what you can imagine what happens is if you have a new farm that's been established on the edge of a forest, and there's been some deforestation that's happened and shrunk that habitat for bats and put pressure on them. When those bats get stressed in some other way, so because of a drought or other weather conditions, and they can't access the food and the shelter they need in their forest, they're going to leave and go to the farm that is right on the edge of that forest. And diseases have spilled over before because there are fruit trees adjacent to where the livestock are being kept. And so the bats roost in those fruit trees, they eat the fruit, they spit out what they don't want, and that lands covered in saliva, which has virus in it, for an unsuspecting pig or horse or other domestic animal to eat, get very sick, and infect the next human who comes to take care of them. They can also transmit to intermediate hosts, so to these amplifying hosts, by urinating and dropping stool on them. So again, when they hang out in those fruit trees over where the animals are living. And then bats can also transmit diseases to humans directly when humans eat bats, when we have contact with their aerosols or guano in the course of occupational or recreational activities, through bites and scratches. So that's really just microbats. Megabats don't really do that. Okay, so talked a little bit, sort of a brief, quick run through some really, really big topics. So that was a very quick overview, but I want to make sure we have lots of time for questions, because often, some questions pop up here. Very briefly going to talk about the features of a One Health study. So just to refresh your memory, because this was a couple of minutes ago now, One Health studies, or One Health, is a conceptual framework that views human, animal, and environmental health as interconnected. And so a One Health study really should involve both the human, animal, and the environmental sector. And so to make this more evident and to strengthen reporting for One Health studies, a guideline was created, the COHERE guidelines, which were modeled after the STROBE and the CONSORT guidelines and are indexed in the Equator Network. So if you have an interest in a One Health study, you can take a look at these guidelines, or you just want to know a little bit more about what's involved. But the key is that you sort of take these observational study guidelines and you really examine if they've been applied to all three of these sectors. Okay, so a couple more slides about the COVID and pet study, and then I can take some questions. So now you've got all of the background. So we set out, actually, in a much more naive time. It was, think back to March 2020, and Seattle was the hot spot for COVID transmission in the US. And this was before, maybe the very early days of when transmission was starting in New York. And we thought, wow, we have this really fantastic opportunity to leverage our location and the fact that we're a Center for One Health research and understand the role that pets play in COVID-19. And so we thought we have to get out there right away, because this thing's going to burn through and it's going to go away. And we have an opportunity now to learn something. We will never have that opportunity again. Obviously, 18 months later, we had a bit more time than we thought, but nonetheless, we got underway very quickly. We started talking to Washington State University College of Veterinary Medicine back in March of 2020. 2020 and headed out to go sampling in April of 2020. But the motivation for doing this study was that at the time, the risks and frequency of SARS-CoV-2 interspecies transmission was unclear. We were pretty sure it came from an animal, but we weren't really sure what happens in a household. And this becomes really, really important because, again, if you think back to the pre-COVID period, we were all, leaving our houses every day to go to work while our dogs and cats were staying at home, sort of doing their own thing, getting up to God knows what while we were gone. And then if you were exposed to a COVID case, or you had traveled, or you became COVID positive, all of a sudden you were locked in your house constantly until your quarantine period was up, or your isolation period if you were a case. And if you can, those of us who have pets, if you're at home sort of stuck at your house, your pet is probably going to cuddle up right next to you and spend a lot more time with you. So the concern at the beginning was, if dogs and cats can serve as a reservoir for COVID, we need to know that very quickly so that we can advise people who are isolating or quarantining for COVID to do so separately from their pets, if at all possible. Otherwise, we run the risk of creating a new reservoir for COVID when people take those measures to prevent transmission to other humans. And beyond being relevant to for veterinary health to know if dogs and cats are at risk of getting COVID, we also had a lot of questions coming from veterinary clinics saying, someone's dog has broken their leg and the owner is COVID positive. Can I see this dog? Is that a risk to me? What PPE do I need? So to try to answer these questions, we launched a community-based study of pets that were living with COVID positive people. And I mentioned this is a collaboration between the University of Washington and Washington State University, and we have other partners on this as well, including Seattle-King County Public Health, but the core is really UW and Wazoo, with those of us at UW leading the epidemiology and the Wazoo team leading the laboratory side of it. So we had two specific aims. The first was to estimate the burden of SARS-CoV-2 transmission from affected individuals to pets in their households. And the second was to identify environmental and animal contact variables that are associated with transmission risk. So those of you who have an epi background, as I talk to you more about our study, you're probably going to think, well, this is a cross-sectional study, so we can't know the direction of transmission. We can't know if people are infecting their pets or if it's the other way around, if it's zoonotic or reverse zoonotic. We feel that because we know now that humans have many, many, many times the risk of pets of having COVID, that we're pretty confident that this is people infecting their pets and not the other way around. So this is a household-based study. So what that meant is we recruit and sample by the household. We define a household as a person cohabiting with at least one dog, cat, ferret, or hamster. I have dogs and cats bolded here because while ferrets and hamsters are eligible, we haven't had any enroll yet, or their owners have not enrolled them. We have enrolled dogs and cats, and I'm going to be presenting dog data here. We have the participant does consent and fills out through REDCap, and then completes a questionnaire on REDCap. And then after that, we go to the household and collect samples of their pets. And if there's been a person in the household that tested positive in the last 14 days, we aim to get to the house in the next 48 hours after they enroll. Otherwise, it takes a little bit longer. So the questionnaire includes an animal contact and environmental variables and human and animal clinical variables. And again, as I mentioned, we try to get out very quickly if there's a recent positive. On the animals, we do nasal and throat swabs. We do a rectal swab, and we draw blood. And this on the top right is myself on the left, and one of our other city veterinarians, Katie Cool, on the right, sampling Olive. And so she is wrapped up in what we call a kitty burrito, which is a method of restraint where you use a towel. It's very nice and calming, and cats tend to like it. And this is really important because we're not using any muzzles in this study. So this slide is, this came from Tim Basler at Washington State University, who is our laboratory guru. This is not my field of expertise, so I'm afraid I can't answer many questions here, but I kept this in case there is anybody here today who's laboratory inclined. So I don't think I mentioned this yet actually, but we are doing PCR testing on the swab. So PCR on the nasal, the oropharyngeal and the nasal swab, which is combined together, and PCR testing on the rectal swab, and then antibody testing on the blood of the serum. So the numbers are going to change a little bit as we go forward, and that's because we have interim analyses for the rest of the results I'm going to present were run a little bit before these results. So what you see here is 61 dogs with completed laboratory testing. We actually have more than that now, but it takes a little bit to sort of update all of our data and rerun and output this. So we actually have, Vicki, correct me if I'm wrong, but I think we actually have 3 seropositive dogs now. And what's really interesting here, and this is a little bit speculative, but all of our seropositives on the PCR have come up quite recently. And so that timing, we haven't confirmed any as Delta variant, but that timing sort of coming after Delta variant came into Washington is certainly interesting. But yeah, I believe we have 3 PCR positive dogs now, and we have 24 serology positive, or 24 with antibodies, and then 31 negative and 6 indeterminate. And then these are ELISA ratios. And again, this is a slide coming from our laboratory team. So this is not my area of expertise as much as what's coming. Okay, so some descriptive statistics. As I mentioned, we set out to not only ascertain how often this transmission was happening in households, but also what were some of the drivers of that. And we saw an opportunity in doing this to try to describe what does human animal contact look like in the context of a pet relationship in a household, because to us that had relevance beyond COVID. So we found that specifically to COVID, we found that 31% of dogs had activity outside of the home during their quarantines. That was things like going to a groomer or a dog park or a veterinary clinic. 51% of households took precautions to prevent transmission to their pet. So that includes things like having somebody besides the positive person walk the dog or feed the dog, things like that. And 63% were aware of CDC guidelines to prevent transmission to pets, because CDC has actually published guidelines to stop that human-animal transmission in a household. Human-animal contact, this is sort of broadly relevant beyond COVID, but we found 13% share eating utensils. This is actually dogs sharing with their humans, which is sort of a weird way to think about things, but it's the way the data are organized. 72% of dogs share a bed with their owners, and 89% are allowed on furniture. And so our sampling has been conducted in the counties that surround Seattle and Pullman. But it's mostly been in the Seattle area, so that's important when interpreting these. These statistics would likely look quite different in another setting. But keeping in mind this is Seattle, we had a mean household size of 3.4 residents, almost 2,000 square feet with 3.3 beds, and 85% of dogs had access to outdoor space. So in Seattle, that all kind of fits with a pretty high socioeconomic status. So again, I mentioned that these numbers look a little bit different than what you saw on the laboratory slide, and that's because we ran these analyses earlier than that table was output. We are updating these soon, but haven't updated yet. So at the time we ran these analyses, we had 0 positive PCR. So you can see they're coming in pretty quickly. We ran these earlier in the summer. We had an average of 5.5 weeks from the last diagnosis to the date of sampling, so that likely explains why we're seeing so many more antibody positive than we are PCR positive. And again, at the time that we ran these analyses, we had 43% positive on serology, and we had a third outcome beyond the laboratory outcomes called COVID-like illness. And so there was a question in our survey that asked if the pet had I think it was a change in appetite or energy level, or coughing or difficulty breathing in the 2 weeks after the owner's diagnosis, or the person's diagnosis. And we found about a quarter of pets were sick in that time window. So our second aim, as I mentioned, was to do some hypothesis testing. So not just to describe how frequently SARS-CoV-2 is going back and forth within households between people and their pets, and to describe what human-animal contact looks like, but also to test some hypotheses we had about risk factors for transmissions. So we had these 3 outcomes: being antibody positive, being PCR positive, or
We lost you again, Julianne.
Just give her a sec to maybe pop back in.
Mid-thought.
She's still active, so hopefully she'll pop back in.
Nope.
Oh,
There she is.
oh, here she is. It looks like you're on mute too.
Oh, I think it automatically muted me when I came back in. Okay. We are almost there. So close. Where did you lose me?
Going through the 3 outcomes.
The 3 outcomes. Okay. So our 3 outcomes were antibody positive, PCR positive, and this COVID-19 like illness. So being sick was something that looked like it could be COVID. And then our risk factors, which we call exposures, are the pet being indoor only, meaning they don't go outside, residing in a condo or an apartment as opposed to a single family house, the square footage of the house, whether the pet shares a bed with people, whether they share eating utensils with people, whether the people in the household took measures to prevent transmission to the pets, so those things like having somebody else who's not positive walk the dog, feed the dog, etc. And then finally, how many people in the household were COVID positive. These are our results. These are interim analyses. We're going to have a higher sample size at the end, so we hope this will tighten up some of these confidence intervals. But the only exposures or risk factors that had a consistent direction of effect across both COVID-19 like illness and seropositivity were bed sharing, which was a increased risk, the household taking precautions to prevent transmission to the pet, which decreased risk, and the number of people in the household that were positive, which increased risk. And I should mention the reason that PCR positive is not on this table is because when I ran this analysis, we did not have any at that time. So what we are concluding based on the interim analyses is that SARS-CoV-2 is actually quite frequently transmitted within households, and that is based on our serology results and I think our PCR results that we're getting in the last couple of weeks are really backing that up. That close human-animal contact is common in this population and may increase risk, and that people in this population, which again is a pretty well-off population, mostly located around Seattle, but they're generally aware of and willing to adopt measures to protect their pets from infection, and that furthermore, these measures may actually be successful. Okay, so I'm ready for questions.
Let me pop open that chat. Vicki, did you want to go through questions or would you like me to?
Maybe you can, so I can keep writing things in this.
Happy to, yeah. All right, I know we got one early on, so I'm gonna scroll back up. So this first one is from R. Lorenzana. It says, has a transmission route of bat-derived fruit pulp to domestic animals been documented?
So, has somebody followed like a molecular signal for a piece of fruit? I don't believe so, but yes, we know that we are very, very convinced that that is how Nipah virus emerged, and that is how Nipah virus emerged in Malaysia and how Hendra virus emerged in Australia. And those are the kind of pig and the horse examples I kept mentioning. Nipah emerged from bats to pigs in that way, and Hendra emerged from bats to horses in that way. And that is how Hendra in particular, people that have direct contact with bats really don't seem to get sick. It really seems like it has to come from horses. So all of the people who've gotten sick and died of Hendra have been from horses, and the horses getting infected from bats roosting above, I think it was apple trees, but roosting in trees above where the horses are.
Oh, it's super interesting that they're not getting it directly from the bats.
Mm-hmm. Bats don't always, it's very difficult to nail down where the bats are that are the riskiest and what makes them risky, because they don't, even these viruses they harbor for a long time, they don't shed them constantly. They shed them in times when they're stressed. So things like droughts and land use changes and things like that cause them stress will make them actually shed their viruses more. So we tend to see Ebola outbreaks tend to tie to droughts.
The next question says, how different are the PCR tests for pets versus human ones?
Ooh, that is a question for Tim, our PCR guru. I think I actually didn't put my email address on here, but I can enter it in the chat, and any really critical questions I can forward to Tim. I believe they are quite different based on how much effort and time it has taken Tim to develop them. In part, I think that reflects that we have a lot of we have endemic coronaviruses in cats and dogs, so we had to make sure we weren't picking up on those instead, in particular in cats. Yes, I believe that they're quite different, but I can't speak to much more than that.
I see Vicky also. I missed, you put an answer in the chat too, so there's a little more detail in there on that.
Basically just said what you said.
I had thrown a question in there. I was asking whether you're able to control if people kind of kept the same behavior as they usually would with their pets, because I'm assuming, we all love our animals. So if we've heard in the news it's possible, we might distance. But I saw you answered that. Do you have anything more you want to say kind of about how you're able to control for that or how that impacted the research?
Yeah, so we didn't control for it in the sense that we didn't adjust in other models for people taking precautions, just because our sample size isn't quite large enough to do that. But yeah, you're absolutely right that, if people find out they're COVID positive and then they immediately separate from their pet, we're going to pick up on that when we test for that precaution being taken as a risk factor, but it might make us less able to pick up on like bed sharing as a risk factor or something if they stop doing that once they're positive. Yeah, I mean, we did uncover that in this population in Seattle that enrolls in a study that is focused on themselves and their pets, that they are aware of and willing to do things to stop transmission to their animals. But we obviously, many people are not able to do that. People that live alone, it's certainly very hard to do. And people who live in a very, a studio apartment, and we have sample people in studio apartments, and not a lot to be done there, short of send the animal to somebody else, which people might not be comfortable with, taking care of a pet from a COVID positive house.
Yeah, makes sense. So the next question is, do you have any data on outcomes for infected pets, such as symptomatic illness, death, or et cetera?
So we didn't actually ask about death, which was something that we probably should have done. That wasn't even really on our radar. So we have that COVID-19 like illness, but we didn't ask about hospitalization or death. Like I said, we are seeing about 25% with that COVID-19 like illness. I can say, even though we didn't ask about it, I don't believe any pets have. I think all of them have recovered uneventfully. Some have had to seek veterinary care. So we have had some enter the study through the veterinary clinic, and we have had at least one hospitalized, possibly a couple more, just based on talking to the owners, but we didn't systematically collect those data.
And then I see a question here, Pavitra, let me know if I'm misinterpreting this, but it looks like it's asking, were all the antibody positive animals from households where there was severe illness in the human who transmitted it?
So we didn't actually, not very many of our very many of our participants had severe illness. We had thought about examining that as a risk factor, but we just didn't have, very few of them were hospitalized. So that was something that was interesting. But the more severe there's sort of a sweet spot there, right? Because if they're hospitalized, then they're not exposed to their pet during that time. So someone who's, symptomatic, but at home seems like it might be the biggest risk to the pet. That's a good point. We actually haven't tested whether they are symptomatic or not as a risk factor.
Just with more severe cases, maybe you don't have the ability to kind of enroll yourself in this as well, if you're kind of getting that extra level of care.
Yeah, thanks for that. I was also wondering whether, on a related note, whether it's possible to reach out to shelters, because a lot of the pets that get surrendered, if a person goes to hospital or passes away, they get, they go via animal control to a local shelter, and and they do go into quarantine for, I think, 10 days or something like that when they know that there's been an exposure. So I wonder if there's ability to somehow, quote unquote, enroll those into this.
We talked about that at the beginning, and then we had a couple more partners, and then it was sort of decided that the study would split, and there would be a household side and a shelter side. And so the shelter side is being led by the University of Wisconsin School of Veterinary Medicine, because they actually have relationships with some of the shelters here in Seattle. And I'm not actually sure where they are in their data collection, but that was something we were thinking about as well. So we decided we would stick with the household, so the people and the pets that are the pets are staying in the household. But yeah, you're absolutely right. And early in the pandemic, again, when we really didn't know if dogs and cats were a huge risk factor for people, this occupational health of shelter staff were a big concern for us as well. So yeah, excellent point there.
And another reason we chose to split it off is because we didn't have the opportunity to get the epidemiological data from the households prior to surrender. So we did not know if it was like, there was a concern actually at the beginning of us doing the study, because there was a concern that people would freak out and drop off their animals at the shelter so that they would not get COVID from the animals. when it was really early on and no one knew what the situation was. So we didn't know if people were surrendering it early on because of fear of that incorrect information of the zoonoses going in that direction, or whether someone was sick or that the owner had died, and that the epi data about contact would have been lost with that.
But it's an excellent point that of all of those COVID hospitalizations and deaths, many of those people have pets, and those that live alone need somebody else to take care of their pets.
Yeah, thankfully that deluge of animals never came. But,
yes.
So.
Yeah, absolutely. So Will asks, is also curious if there are any data about incubation period in dogs and cats, and if you have any plans to compare transmissibility data from unvaccinated infected humans versus humans with breakthrough cases.
Yeah, so to answer your first question, there might be some from the laboratory data on incubation. I don't think in dogs. Cats have been used a little bit more as a laboratory model, but I'm not actually certain what's come out of that. I haven't seen that, but I know there have been some cats experimentally infected with COVID. So that would be, that's one thing. Those of you, I think probably quite a few of you are veterinarians in this call. Those of you who are not, there are things you can expose an animal to that you cannot do in human subjects research. So we can do things like infecting within the confines of the ethics of animal research. We can expose a cat to COVID and observe what happens in an experimental fashion. I'm not sure what the incubation period is. We've been operating there under the assumption it's pretty similar to humans. And then the second question was the role of vaccines, and we are actually working on a grant to try to do just that.
Oh, exciting. Well, if you get it, we'll have to have you back. Ellen asks a similar question. Given that it is not entirely known the extent of transmissibility in fully vaccinated individuals, is it possible basically that a fully vaccinated human can still transmit to a pet?
I don't see any reason that they couldn't. Yeah, absolutely. I think that they could. And that's why we've been really interested in continuing to study pets as vaccines roll out, especially because there's no dog or cat COVID vaccine, so they don't have an vaccine.
A follow-up question, I guess, to that is: can you postulate? Do you foresee potentially a vaccine for cats and dogs going forward?
So there is a vaccine. So mink are being vaccinated, and I think actually, I read recently that the LSU tiger, Mike the tiger at LSU, the LSU mascot, is getting vaccinated. So there has been some off-label use of the human vaccine in animals, and there is an approved vaccine for I think ferrets as well, and mink. Or perhaps it's just for mink. My thought is, and this is not the area where I'm an expert, but my thought is that we would need to demonstrate that pets are transmitting to people, or there would need to be evidence that it makes pets sick, and more than a sort of self-limiting. They don't want to eat their food for a couple of days, but then they're fine. An illness that's more severe than that, for there to be the financial incentive to develop a vaccine.
That makes sense. Vicki, I thought I saw you on mute earlier. I just wasn't sure if maybe I cut you off or.
I think I just remuted myself if my children were running through the house.
Oh, good. It looks like that was the last question to come through. If anyone has another question, go ahead and type it in. We'll just give a virtual minute here in case something else comes through, if you want to raise your hand and unmute. Otherwise, this has been so educational, just very interesting. So thank you guys for the work you're doing, and we're excited to kind of follow along and see what information you glean over the next however long we're still going. Do you have any ideas about how long you'll be able to continue conducting this study? Do you have an end date?
We want to sample 30 more households. So It depends on how long it takes us to get to those households, to enroll those households. And then because, we're going out, we have a team going out to people's households. It requires, even if 30 people were to enroll next week, it would take us a couple of weeks to be able to get out and sample all those households.
But we are still recruiting, and it's very easy for you to participate in the study. It's just like a 10-minute survey. It goes super fast, and then our team comes by for about 20 minutes, or depending on how many animals you have, but it's really short. So if you or anyone you know has recently had COVID, we are really trying to get people who are actively shedding virus themselves right now. But we will come out and sample your animals if it's been a couple weeks too, even if you've fully recovered. But I put the link in the chat, but RedCap is down today. I don't know why, but it should be up and running again soon. And that link I sends you to a webpage where you can learn more about study and enroll.
Yeah, so it's very easy on your end. It requires a little bit of logistics for us to get out and sample households, but on your end, it's very, very simple. And then there was one last request for a takeaway at the end. And so takeaway that I would give, and this has actually been found, and there have been a couple of other studies similar to ours going on in different parts of the country and in Canada as well. It looks like we are seeing quite a bit of transmission from humans to animals in their household, and it seems like a not insignificant proportion of those animals do have some degree of illness, but that illness appears to be self-limiting. So in most cases, they don't need to see a veterinarian, and they certainly don't die or need to be on a ventilator or anything like that. A couple of them have been sicker, but that seems uncommon. And it does look like the CDC guidelines to prevent transmission to pets does reduce the risk of transmission. So those would be our takeaways. And I see Vicki answered the cat questions. So yes, stay tuned for a paper that we hope will be out in the next couple of months that should have both dogs and cats in it.
All right, well, thank you both so much. As you can see, everyone, we are recording, so we'll go ahead and get this up onto our YouTube. So if you're thinking of someone who you think would really have enjoyed it, just go ahead and give us about a few weeks. We'll get it up on the whole YouTube, so anyone can tune in. Again, thank you so much, Julianne and Vicki. Thanks for your research and for taking time to be here today.
Thanks, everyone. Have a good afternoon.